to the lecture series in cell culture. So, we are into the 7th week, and as of now we have using a case study of neurons, we have talked about how to separate different kind of neurons, and two processes what we have discussed in depth, which are kind of can be followed by any lab at any point of time, is density gradient centrifugation. And this density gradient centrifugation, this process using sucrose or nicodens or optiprep as the material to make the density gradients. This technique can be used for cell separation of pancreatic cells, this could be used for the liver cells, this could be used for any cell type, provided there are a few prerequisites to that, provided you obtain a single cell suspension. What does that mean? That means, so whenever we take out a chunk of a tissue, so it consists of millions of cells. Okay. So, now you have to have a way by virtue of which you use some form of enzymatic or mechanical or some treatment where individual cell dissociate out from the tissue. So, it no more maintains the tissue architecture, instead it becomes a single cell suspension, something like this. I mean, just start off with. So, this is your week 7, lecture 1. Okay. So, say for example, you have this piece of tissue what you have to dissociate, and these are the individual cells. So, for example, something like a 3-dimensional tissue. Okay. Okay. Now, if I have to use this, so say for example, I say I have certain cells which are in orange, certain cells which are green, and these are different cell types based on their different functionality. Okay. Some cells which are in yellow, like this. This is how the, and you want to separate all these different cell types. So, the in order to use, whether you use so we have talked about two techniques, right? We have talked about density gradient, and we have talked about immunopanning, where you are using an immune marker. Now, for both these techniques, what is very essential is that from this mass you get single cell suspension, and this single cell suspension means essentially if you dissociate this tissue, okay, if you dissociate the tissue, what you will be getting is something like this. One second. Okay, something like this. And one minute, let me come to. So, now if you get this kind of mix and the individual cells have different size, then by virtue of which you can follow the density gradient. provided each one of these population have different densities to follow. Now, say for example, they do not have different densities to follow, then if they have different kind of immune markers, something like this, and you have an idea that these are the immune markers which are present there. Okay. So, then what you can do, then you can follow something called an immunopanning, what we have discussed in the last class. Further, an extension of the technique is cell sorting. You can use FACS, which is fluorescent assisted cell sorting. Okay. FACS, which is essentially, F stands for fluorescent, A stand for assisted, C stand for cell, and S stand for sorting. This is another modern technique. So, what is really FACS? So, we have talked about two. So, now I am adding a third frame to this, which is your FACS. What really FACS is all about. So, we will discuss a little bit about in this class about what is FACS. So, even before getting into any kind of a study on FACS, try to understand the basic fundamental logic, what is happening. So, say for example, you are going to a stadium to see a match or something. So, there are say 1,000 people, and each one of you or each one of there are. So, say for example, I divide that VIP gallery, semi VIP gallery, then I say this is only exclusively reserved for families of some big giants, and then I reserve section for some sportsmen or something, then I say this is a general. So, I have 5 different categories. Now, when I am entering through one common gate, right, but the difference. So, say for example, this is the common gate through which I am entering, and this is the arena where we are supposed to go, and here people are coming of different types, okay. And these are the different kind of people coming to view the match. Now, say for example, out here, out at this place, I have a sensor out here. So, each one of these people are coming. So, the sensor will tell, okay, this is blue, light blue. Okay. So, these light blue should be asked to move to this spot. This is the arena. Okay. Now, this is red. Okay. This red should be asked to, this is reserved for the red should move here. These are greens, the greens should be here, and these are say pink should be here. These are yellow should be sitting here. Okay. These are light say orange should be sitting here. Okay. So, what I wanted to say is, say for example, on the cell surface you have specific markers. If you consider these as different cells, instead of these are individuals who are holding different color tag of tickets. So, they have this ticket for it. Okay. So, I have this color. So, okay, for you this place is reserved. Okay. So, for light blue this place is, for light green this is reserved, for pink this is reserved, for yellow this part is reserved, for red this part is reserved. Okay. So, there are locations which are reserved. Now, but you have to location is not an issue. Issue is how you separate them out. That is the most critical point. So, in order to separate them out, the first thing what one has to do, if you get these specific cell types, say for example, I get this, I get this, I get this, I get this, I get this. So, there are 5 cell types. Say for example, I am getting A, B, C, D, E. So, I told you one options I have is that I separate them out by density gradient, which is possible actually. But if they are of the same size, then the density gradient won't work out, because the density of the two particles, say for example, I just introduce another one, say F. Now, between D and F, you hardly will see a density difference, or similarly between A and B, there will be very little option that there will be a huge density difference. You understand? So, in this situation, say for so now what you have these two population, this two population, which will behave differently as compared to this population. Now, how to separate them out? Now, one option is that you label them, just like you are entering the arena to see a match. You have a specific kind of color coded tickets. You may have an orange one, you may have a green one, you may have a blue one, you may have a red one. Now, based on that code, here there are sentries who are standing out here. They will tell you that, okay, your sitting spot is here, or your sitting spot is here, or yours is here, or yours is here, or yours is here, or here, or here, wherever. Okay. Now, at the microscopic level, these sentries will be replaced by specific probes. And the kind of probe are we talking about is called fluorescent probes. The probe which will distinguish one cell from another based on the fluorescent signature, which either the cell has an inherent fluorescent signature, or you introduce a fluorescent label on the cell. So, say for example, you know you have a mixture like this, a mixture of cells like this. Now, go back to that previous diagram where we started. So, you know this is your tissue has this kind, it may be a pancreatic tissue, it could be a liver tissue, it could be a spinal cord, it does not matter, that is irrespective. Okay. I am taking examples of neurons because I have done intense work on that area. So, I know exactly how, but the logics, it is not that from the neuronal book I picked up the logic. Logics were standard cell culture logic, but then you have to take a cell type to tell you, and based on your situation you will have to pick up the conditions, but you have to understand what all basic tools you have. So, this whole purpose of this exercise is you know what all scientific or cell culture tools you are having at your disposal in order to crack a problem, right. So, coming back, so if you look at it, so say for example, how many colors we have? We have, say we have blue, and these colors are very symbolic here, it's nothing to do with real life colors. Red, you have green, and you have orange, and you have yellow. Now, suppose you know these, you have these 5 different kind of cells, and you know there are unique antibodies which could be bound on their surface, which, say for example, I take, so now I label the different antibodies, antibody B, antibody R, antibody G, just sign that, this sign is saying that this is an antibody, okay, G, O prime, Y prime, okay. Now, if I bring Y prime into the game, I know only yellow cells, it will adhere to the yellow cells, right, no issues. Now, I bring, so now these cells will have a tag Y prime. Now, what I have to do, I may, if this is a primary antibody, I have to have a fluorescent tag which will distinguish these cells. So, say for example, I want to separate from all these 5 different cell type, I wanted to separate 2 cell type, just hypothetical say, the green and the yellow ones. So, what I do, I put a G prime. So, now I have 2 cells which are now labeled with 2 fluorescent probes, yellow and the green, right. Now, I have a sensor, say for example, I allow the cells to flow. So, I have something like this, a very narrow tube through which only one cell at a time can flow. Okay. And here I have a sensor. And here I have a gate. Now, I have multiple cells which are coming through, right? And yet I have, actually this drawing had slight mistake and I am just putting it now. Now I am putting it. Okay. Now I am getting cells of these kinds. So the yellow, right? And at a time only one cell is coming through. Red, red, pink, again a yellow, yellow, and you have the green likewise. Right. Now, here you are having the probe. Probe to distinguish, in this case study, okay, probe to distinguish between green and yellow fluorescent labels. Okay. So this channel, so we are having two channels now, right. So this is one channel which I call this as a yellow channel, and there is another channel which we call this as a green channel, because this is what I am going to separate out, and this is a non-specific channel, right. Now, what will happen once the cell will approach here, say for example a green cell approaches here. Now, as soon as this sensor senses this, it will send a command signal which will remove this. So, okay, by the way, there is a third gate here. So, it will immediately open this gate, and the next phase what you will see, this will remain closed, but this green gate is now open, and this cell will move. So, now you have a collection vessel where you are collecting all the green ones. Similarly, if there is a yellow one coming through, so what will happen in real life? Now, a yellow cell coming, this gate will remain closed, this will open and the yellow cell will move here, and you can collect all the yellow cells in your bucket. So, now from 5 cell types, now you are slowly narrowing down to 2 different cell types. So, you see this is how, so this fluorescent sorter, so depending on how powerful is your sorter. So, this is your fluorescent sorter. So, it is the same analogy what I told you in when you enter a stadium. So, here you have the sentries standing there who are ensuring which part of the stadium you should go. So, these fluorescent cell sorter, fluorescent assisted cell sorter are one of the very potential tools in the modern cell culture labs, and this comes very handy when you have a complex situation like this, where you have multiple cell types to be separated out, but the fact is still the same what I showed you in the last class, that you have to have those unique markers. Without this, the basic fundamental is still the same. You have to have those basic markers. Without that, really to sort them out won't be an easy job. Okay. So, now in the light of this, so we have density gradient centrifugation, you have immunopanning, you have fluorescent assisted cell sorter. So, we have three different techniques by virtue of which one can do a cell separation. And now, in the light of this, if you go back and think what all we covered, think over it. You have different kind of neurons, different kind of oligos, microglia, astrocytes, and not only you can separate them out using density gradient, you can separate them out further. Separation can be achieved by virtue of which they are immunopanning. As a matter of fact, you'll be surprised to know regarding this immunopanning, if you really know at what point of time these specific cell express unique markers on their surface, all this kind of fluorescent cell sorting can be a really very formidable technique by virtue of which you can isolate them. Okay. So, I will close in here for this class. In the next class, we will go further and discuss about some of the other aspects which will help to have a much more real life model of cell culture. Thank you.